Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have a returning guest, Nils G Walter. He's a professor of chemistry and biological chemistry as well. We spoke once a while ago about various types of RNAs that are in our cells. It was a really interesting interview. I'm having him back to be one of the co-authors of the book on viruses I'm doing, because again, I think his perspective and the stuff he studies about RNA is very rare compared to a lot of the people I've spoken to. So I want his input. So Nils, thanks for coming back. How you doing?
I'm doing very well. Thanks for having me, Richard.
First, I want to get your bio again. So tell me about your career a bit and then your current research.
Right. So I was trained in Germany as a chemist with emphasis on biochemistry. I got my undergrad degree, my master's degree, or at the time it was called diploma, and my PhD all in Germany. My PhD, I worked at the Max Planck Institute for Biophysical Chemistry, which at the time had a Nobel Laureate as the director, or as one of the directors. And I worked with him directly, Manfred Eigen. He actually passed away just last year. And he actually worked on in vitro evolution. So in vitro evolution is the idea that you can have a Darwinian selection, a process that would mimic how life works with replication, mutation, and selection from the diverse sequences you get through the mutation out of that replication. And through this Darwinian evolution and cycles of it, you eventually can evolve a new function. And so that attracted me to his lab. That's why I joined his lab. I actually was the biochemist responsible for building an in vitro evolution machine that then later apparently went to the Technical Museum in Munich as a museum piece. But what I learned from it is understanding that molecules, just as viruses, can actually evolve over time. And there is a connection, as you can tell, between in vitro evolution and what we now see has emerged as a virus, coronavirus or SARS-CoV-2. And that, of course, is fascinating to see that connection. Now, after my PhD there on in vitro evolution, I moved to the US as a postdoctoral fellow at the University of Vermont, so a less traveled path. And I learned about RNA enzymes, or ribozymes for short. They are connected to in vitro evolution because they could have been the starting point for how life formed on Earth, because RNA is a unique molecule across biology that has a feature of carrying genetic information like it is in the genome of the coronavirus, of the COVID-19 agent, but also can act as an enzyme like proteins are thought to do. And so it kind of bridges DNA and protein in a way. And as such, solves the chicken and egg problem, basically allows thoughts about how life on Earth could have started, because there could have been an RNA world where only these RNA enzymes, these ribozymes existed that made themselves ultimately and replicated and mutated and evolved over time. And it's these ribozymes that I studied as a postdoc. And that's how I started my own lab then in 1999 at the University of Michigan, where I'm still today. I'm in chemistry, but as you, Richard, said, I have multiple appointments in biophysics, biological chemistry, and across campus. I founded a Center for RNA Biomedicine, which is studying RNA with the hope to derive medicines from it, either something like an RNA-based vaccine, as we are seeing in clinical trials against COVID-19 nowadays, or to use RNA as a potential drug target to cure diseases, and there are examples for that as well. Very famous SMA, or spinal muscular atrophy, can now be fixed with basically RNA, a modified RNA molecule, if you wish, that fixes what we call a splicing defect and helps the toddlers that have SMA survive into teenage years so far. And so there are clearly abilities to utilize our knowledge on RNA to cure diseases. And the Center for RNA Biomedicine that I founded and co-direct, has this goal to bring people together, researchers from different areas, different disciplines together to utilize the power of RNA for medicine down the road. And so that's what I do today.
Got it. Okay. As for the questions now that we're going to get to, is there any form of life that you know of that doesn't have a virus that preys upon it or interacts with it?
Very unlikely. And the reason is there is, of course, a co-evolution, if you wish, between a cellular life form. So kingdoms are archaea, bacteria, and eukaryotic cells, so cells like in our body with the nucleus and the cytoplasm. In bacteria and archaea, you have the two combined. You have only one compartment, one membrane around the entire cell. And viruses have evolved. The viruses that we see today are relatively modern in origin and have evolved together with these cells. So as soon as there was the first cell, probably there came to exist the first virus that would have infected that cell. Every so often, it will succeed in killing the cell. But if it does that, then of course its host is dead, and the virus itself that needs the machinery of the cell to survive itself as a parasite will also have vanished. So in the end, it's best for a virus to kill maybe some of the target cells, but not all of them. And in that way, over time, over eons, over billions of years, really, viruses and single cells and later multicellular organisms like the human body have co-evolved to coexist. And I think there will not be, because of that ancient origin of viruses, there will unlikely be any organism today that doesn't have a virus associated with it.
And going back to origins, do you think that viruses arrived on the scene before cells, or coincidentally, or contemporaneously? What's your thought?
Yeah, so that's an interesting question and somewhat philosophical and certainly also difficult to answer in a way. The way I think about this is that a virus itself, the difference between a virus and a cell is that the virus lacks key components of what we call metabolism, of being able to produce all the molecules that it needs to be a life form. And the virus becomes a parasite, or is a parasite that only can exist and replicate in a cell that has these missing components, the components that are missing from the virus. And so for that reason, the very definition of a virus is that it has many components that are important. It has a replicase enzyme that can copy its own genome, be it RNA or DNA. It has the DNA or RNA. It has some sort of a coat protein. It has maybe membrane around it like a cell has, but it lacks other components that are necessary to make, for example, the building blocks to make the DNA or RNA genome, to make the protein that is then the replicase enzyme. And because this metabolism is lacking, the virus can only live inside a cell. Outside a cell, the form that it takes there is, again, like we've seen with the COVID-19 agent, a SARS-CoV-2 virus, an RNA that wraps itself in a protein coat, that wraps itself in a membrane and has little spikes on it and other proteins, but ultimately in itself cannot replicate without the help of a cell. So in a cell, in a way, as a parasite, it subjugates the metabolism of the cell to make more copies of itself. And now it comes down to whether you feel that the ability to actually replicate, so not just the inherent ability, but the actual factual possibility to make itself in many more copies of itself is part of being alive or not, that then distinguishes whether you would say a virus is alive. I would say it's alive inside the cell, and it basically is not alive outside the cell because it cannot replicate on its own. It needs the cell as a helper to do that.
Yeah, that's good. So I guess contingently alive. I want to give you an example I thought about and see if you think it applies. You know about trees, and let's say certain ones come from seeds, okay? If most of my life I see seeds and I rarely see a tree, I'm going to think, oh, trees are not really alive. If they're in the right maybe soil and sunlight and all that, then they're alive. But most of the time I see them, they're seeds and they're not alive in that point, or they're dormant. And I think that possibly may be what's happening with viruses. People, I think, contemplate them in the virion stage mostly and don't in the intracellular stage. And therefore, I think that's why they say, oh, well, virions are non-motile and they're just passive, and the virus can't exist unless it's inside a cell, and therefore it's not alive, or at best contingently alive inside a cell. But what's your thoughts on that example? Do you think that applies?
Yeah, again, I think it's somewhere in between. One way to think about it is, there are viruses that get incorporated into the genome of a cell. In fact, 8% of the human genome is retroviral in origin. That is, it comes from viral particles, virions, that at some point infected a eukaryotic cell, an ancestor cell, and got itself integrated in the form of DNA into the genome that we now carry with us. So these 8%, which is thousands of different viral infections, were at some point experienced over the millions of generations that brought us about, and accumulated over time in the genome, and there was more and more of this buildup of retroviral RNA. However, in order to be silenced and not just break out and destroy the cell, which would be unproductive for the virus as well as the cell, these viruses became silenced. There's a whole machinery inside the cell that silences these viruses. And at some point, they also mutated and nowadays are no longer fully functional viruses. But the genome is clearly highly related to something like HIV virus, something that has the ability to replicate inside the cell and then integrate itself into the genome. So that is well known. There is also evidence, or there's a lot of thought about the idea that viruses, when they infect and replicate in the cell, can actually take snippets of genetic information out of the cell with them and co-opt certain genes that might have evolved in the cell that it infected, and just by chance, basically, through, if you wish, a mutation, splices itself into the viral genome and then gets carried on. If it's not helpful to the virus will eventually just discard it, will lose it over subsequent cycles of infecting cells, replicating. But if it is useful, then it will carry that gene with it onward and will be able to now adopt it into its own genome for the long haul. So in other words, there is evidence that viruses can insert into our genome, but there's also evidence, or high likelihood at least, that pieces of genetic information from our cells also can move into viruses. In fact, there's a whole group of so-called transposons that are retroviral-like, that are known to hop from one cell to another, from one organism to another. And of course, through that process, the virus enriches itself in its genetic makeup and can, for example, adopt a new protein that, in the case of SARS-CoV-2, eventually evolved into a way that it could hook onto the ACE2 receptor on human cells that are found in a nasal passage, in the kidneys and elsewhere, intestines, and replicate so efficiently as we see today in this pandemic. So it's really a co-evolution between the two.
Yeah, I see you got a lot of reason why viruses appear to have, I'm going to call it agency and evaluation of ambiguous information and contingent action inside of a cell. It's amazing. I mean, some of the things you're talking about, I'd like to explore with you at some point, but yeah, it's unbelievable. When you contemplate extracellular vesicles or plasmids from bacteria or things like that, do they seem viral-like to you? You know, they carry genetic material, they can enter into cells, they can affect gene expression, etc. What's your thoughts there?
Yeah, so we actually work on a protein called ARC, which is found expressed in neurons, in cells of the brain. And this ARC protein resembles certain code proteins of retroviruses, again, like the AIDS virus, HIV, or other similar viruses. And in the virus, it has the function, and it's the same what it does as an ARC protein in our cells, to form little vehicles, basically more or less spherical entities that can bud from a cell and take some cargo with them. So in the virus, right, where you have this as a code protein, it helps the virus, after it has replicated and made this protein in the cell, to package its own genome, make a little sphere around it, the virion, and then escape the cell, take some membrane with it as an envelope around the virion, and then pass to the next cell that it can infect. But it turns out that this ART protein that is highly homologous, very closely related to these retroviral packaging proteins, does something similar in our neurons, where it also can take genetic information from inside the cell, but out from the cell, and then transfer this genetic information to a neighboring cell. And it's thought that that may help directionally transfer genetic information from one cell to the other in a way where they communicate in a material way, where transporting genetic information from one cell to the other, reprogramming the recipient cell.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And so just like